As she mentioned, my name is Angela Tom. Thank you for welcoming me to Hunter's Hope to present on Orchard Therapy's platform, and we'll talk a little bit about that as we go through the slides. I am the field director for U.S. Medical Affairs and lead the field medical team in engagement with providers in the United States. I work in collaboration with Jenna Swan. She is our patient advocacy lead and help to coordinate this particular presentation. So I want to shout out to Jenna to thank her. Most of all, to thank any of the patients and families that are listening today. Next slide. Orchard is a global gene therapy leader in hematopoietic stem cell gene therapy. We're focused on treating rare inherited disorders where the disease burden on children, families, and caregivers is immense, and where current therapeutic options are limited or just don't exist at all. Next slide. So there's a lot of words on this slide, and I'll spend a few minutes running through it with you. Orchard used the same ex vivo autologous hematopoietic stem cell gene therapy approach for all of its programs. And we're going to spend time today walking through what that looks like from a patient experience perspective. But first, we need to explain what ex vivo autologous HSC gene therapy means. So, hematopoietic stem cells, or HSCs, are found in bone marrow and are responsible for all blood cell production. That includes white cells, red cells, and platelets. HSCs reproduce copies of themselves, known as self-renewal, to sustain blood cell production throughout life. The throughout life point is pretty important when we're thinking about this gene therapy platform. A gene is the DNA code for a specific protein. And the protein is made by turning on or activating a gene when the cell needs that protein. When a gene contains a change to the DNA sequence, this is a mutation or a variant, it does not make that functional protein, and a genetic disorder may arise. In gene therapy, we insert a functional or working copy of the gene to make the missing protein or dysfunctional protein. We do this ex vivo or outside the body by harvesting cells from the patient that are sent to a specialized laboratory. At that laboratory, those HSCs, or hematopoietic stem cells, are isolated and then they're genetically modified before being administered back to the patient. Next slide. So ex vivo autologous HSC gene therapy is a mouthful, but it offers a highly differentiated approach. This is a one-time administration that provides the potential for a permanent disease correction. Remember, we mentioned that the bone marrow produces cells for a lifetime. So in the upper left corner, you'll see where the green sort of diagram is the lentiviral vector. This inserts a working copy of the gene permanently into those HSC or stem cells. These genetically modified cells can lead to multiple corrected cell types. So these genetically modified cells renew themselves or reproduce themselves, and then they differentiate or they become all the different types of blood cells, red blood cells, platelets, tissue macrophages. And these gene-corrected cells can address multiple organ systems. And because they can address multiple organ systems, they have the potential to treat a variety of disease states. Next slide. I'm going to take some time to walk you through the process from the patient's perspective over the next several slides, and then we'll go through it again with a timeline attached. So following a pre-screening by the doctor, the patients travel to a qualified treatment center where their cells are collected. And then that patient can return home while the cells are shipped to a manufacturing facility. The cells are the stem cells, the blood cells that are needed. At the manufacturing facility, several steps occur. First in the steps is isolation of the needed stem cells. Next slide. Then a functioning copy of the non-working gene is inserted into the cells. So this is known as transduction, with the vector. There's then a need to quality control and test this to make sure it's a safe product, right? The cells are safe, the cells are living, the cells are replicating, they are doing what they're supposed to be doing. The cells are cryopreserved to allow time for testing and shipping back to the treatment center. This could be a long distance. It's important that we make sure the cells are going to be stable. Once the modified cells are received back at the treatment center, that's when the patient can come back in and undergo a conditioning. Conditioning medicines create room in the bone marrow for the gene modified cells to engraft and grow. Next slide. After the conditioning regimen is complete, the gene modified cells are delivered to the patient via intravenous infusion. The patient will remain in the hospital to allow them time to recover from the conditioning regimen and allow time for those cells to engraft and grow. Following recovery from this treatment phase, the patient can go home and be monitored as an outpatient. Next slide. So here's a timeline attached to that same process. If you'll follow with me from the upper right-hand corner, there's a box that shows pre-collection activities. So the physician taking care of the patient is going to make sure that this is the right therapy for that patient. It's really important. Once the physician has determined this is the right therapy and referred them in for the treatment, they'll go to the treatment center where the cells are collected. The cell collection can take anywhere from 2 to 3 days up to 7 days, depending on the way the cells are collected. They can be collected by bone marrow harvest, which is a needle into the bone to extract the marrow, or by mobilized peripheral blood collection, which requires some medication to stimulate blood cells to be generated and then removal from an intravenous route. The cells then are sent to the manufacturing facility. The patient at this time is at home, managing as they would otherwise manage. At the manufacturing facility, the time for this collection, transduction, and quality control cryopreservation process, this could take anywhere from several weeks to a few months. So it could be, depending on the disease state and the need, it could take a few months' time that the patient's at home. Once the treatment center receives the cells back, then the patient is called back in. We want to make sure those cells have safely arrived back before we do anything more with the patient. The patient arrives, they undergo the conditioning medication over several days, depending on the disease state. And then the drug products, the cells that they gave, are readministered. That readministration doesn't take very long. It happens in a single day, and the patient's monitored throughout the infusion. Then the patient needs to recover, and that can take 4 to 8 weeks or so. And it's really based on the clinical condition of the patient. And once the physician that's caring for the patient determines that they are safe to go home, that their immune system has reconstituted, and that their cells that are gene modified are doing what they need to do, then that patient goes home for outpatient monitoring. Next slide. I'm happy to answer questions as we move forward. I know there's a Q&A session. I'd like to reiterate that I'm very grateful to be offered the opportunity to speak with the patient audience, and that I work closely with our patient advocacy lead, Jenna Swan. Thank you so much.